Welcome back to our chapter, or our partial chapter, on carbohydrates. I have to apologize. I'm really busy. I kind of went out on safari. I'm going to close the windows now so we can actually hear. But there may be some noise going on. I'm not sure. I'm going to try to keep us away, but I had to do this. So my crazy schedule. So carbohydrates are really made up of simple sugars or polymers of sugars. Remember, polymer just means lots of them. The monomer we're most interested in is glucose. That's the one we'll focus on, but first we're just going to talk about simple sugars and sugars in general. The way sugars are linked together, or the bonds between them, are called glycosidic bonds or glycosidic linkages. So we've seen so far, if we're talking about sugars, which normally we're talking about glucose, right? That's a glycosidic linkage. If we are talking about amino acids, what's it called? Peptide bond. What about our nucleotides? Yes, you guessed it, phosphodiester. All of these occur through what? Condensation reactions. Ta-da! So what is our telltale sign to figure out what a sugar looks like compared to some of the other monomers that we talked about? Ooh, holy cow. Okay, sorry, that was a close one. Carbohydrates, the telltale sign is carbons, hydrogens, and oxygens. Lots and lots of carbon-hydrogen bonds, but there's always oxygen involved. So there's always oxygen molecules. Oops. Oh my gosh, I can't spell. Oxygen molecules involved with carbon hydrogens. I don't care about these two. You have to deal with that when you get to biochemistry. So we don't care which is which. We don't really care why. But I wanted to show you this picture because the backbone is carbons and there's a bunch of hydrogens and oxygens attached. Okay? Carbons, hydrogens, oxygens. This is very important. These are the telltale signs. Carbon, hydrogen, oxygen. Must have all three. And lots of them. This is a picture of glucose over here in its straight stick formation. Many sugars form ring structures, but we need to know what they look like in their stick formation as well. As you can see, the backbone is all carbons attached to lots of hydrogens and oxygens. Okay, again, this carbonyl at the end, backbone of carbon, hydrogens, but also oxygens stuck onto these carbons. It's really important. Because when we look at the fatty acids, in lipids, let's do a little foreshadowing, lipids only have CH in their backbone. Okay, no oxygen in lipids in the backbone. It's a way to distinguish between this structure, especially the stick structures of sugars, and a long-chain fatty acid. So that is important to know. Whoa. Okay. Hang on. Let's get by this. All right, we made it by. Okay. So ring structures. You need to be able to identify a sugar looking like this: carbons, hydrogens, oxygens, and in the ring structure, which would look like this guy. What else do we see? Carbons, oxygens, hydrogens. These are 6 carbon sugars. What was the sugar in our nucleotides? Uh-oh, nucleotides have a sugar too. We don't want to confuse those with regular sugars that form carbohydrates. The ribose and the deoxyribose in our nucleotides, right, are 5 carbon sugars. You can have 5 carbon sugars. You can have 6 carbon sugars. What do we notice about sugars that, like glucose, or that form polymers into carbs? There's no phosphate group. Do you see a phosphate group anywhere? There's no base. There's no nitrogenous base. Do you see a nitrogenous base anywhere? No. We're going to get to alpha and beta in just a second. Okay, so why do we care? Well, we need sugar. Sugar is energy for our cells. We use sugar to generate ATP. There's lots of different monosaccharides, and these are the essential differences between them. They have unique structures and functions. They're used in different ways for different organisms. Yay. A polysaccharide, or a complex carbohydrate, right? The simplest polysaccharide is just 2. Simple sugars, again, condensation reaction through a glycosidic linkage. Those things are important, just like amino acids, just like the nucleotides. Sugars are linked together through a condensation reaction. We call it a glycosidic linkage or a glycosidic bond. So let's take a look at this. The glycosidic linkage can form essentially in 2 main ways, as an alpha 1,4 and a beta 1,4 glycosidic linkage. Both of them form between the same carbons, blah, blah, blah. Let's look at this instead. Condensation reaction forms a glycosidic linkage, 2-alpha glucose, right? And that's just, again, the orientation of these guys, which I'm not going to ask you whether they're alpha or beta until they make the glycosidic linkage. I will ask you and show you a disaccharide and ask you if it's alpha or beta. So how do we know? How do we know alpha or beta? If you're looking at an alpha, it's the orientation of the bond. And usually they show it in the textbook pointing down, but that's not what you need to look for, because what if you looked at it from the other direction, right? It would be pointing up. So what we need to look at is the orientation of the entire glucose molecule and where the number 6 carbon is. Here's the number 6 carbon, and both are facing up. So I call it 2 thumbs up, because this carbon with the hydroxyl group on it is like a thumb. Now, I have no way of drawing a good thumb here, so I'm not even going to try, but you can think about it. If you hold your hands together, 2 thumbs up. Both of those facing up. If that happens, or both facing down, right? Either one works. So both up or both down has to be alpha 1,4. And they call it 1,4 because it goes between 1 and 4. So it's an alpha 1,4 glycosidic linkage, or glycosidic bond, formed from a condensation reaction that releases water. So if we are looking at a complex carbohydrate, which is linked together by many, where are the glycosidic linkages? Right here. And what are these? Both thumbs up, both thumbs down? Or one up and one down? What do you think? Alpha. These are both up. Has to be alpha 1,4. You need to know this. Need. You need to be able to know it. You need to be able to look at it. And you need to be able to tell me that. This will be on the exam. There's no other way around it. So please make sure you understand these. Shut up out there. These are things you must memorize. You must. Practice. So this one here is a beta 1,4. Why? Because it's got 1 thumb up and 1 thumb down. What do I mean by that? If we again look here, here's our big fat ring structure. Wow, there's some action going on out there. I don't know. We have number 6 up on this side, down on this side, right? Here's the hydroxyl group. This is the number 6. This is the number 6. 1 up, 1 down equals beta 1, 4. So anytime you see 1 up and 1 down, you know it has to be a beta 1, 4 glycosidic linkage. If they're both up, it has to be alpha. That's just the way it is. So I'd like you guys to do that. You can draw an alpha 1,4 glycosidic linkage between two glucose molecules. And again, your glucose molecules can be a pretty, cheesy hexagon, and you don't have to show the thumb up as that actual chemical group, but please show it as something. Draw a thumb there, right? What is alpha? What do the two thumbs look like? Number 10, screenshot, upload. Okay, this one, I'd like you to draw a beta 1, 4. Again, this is number 11, homework upload. These are both in Monday, February 9th, where we were supposed to do the carbs. So please look for them there. Number 11, homework. Number 10, homework. Please do that and enjoy yourself. And thank you for coming. I hope that you enjoyed the safari as much as I did. See you later.